Hello, good evening, Kittu, good evening, Komal Preet, good evening, Achal. Am I audible? Am I audible to everyone? Yeah, okay. So today just I am going to start a live session on basics of biochemistry. We'll see if it works, then we'll go for further also. Right. So wherever you are having questions, you can type in the chat box, and I will try to solve during the lecture only. Fine. So just wait for 2-3 minutes, and then we are going to start. Yeah, not today. In the next coming lecture, we will cover all the biochemistry. Slowly, we will cover it. Just wait for 1 minute, Akshay. We are going to start. Okay. So we are just going to start. So today we will talk about basics of amino acids, their classifications, and then if time permits and you people will be agree, then we will go for physicochemical properties today itself. Otherwise, if we will not have sufficient time, then we will go for another day. Fine. So whenever we are talking about amino acids, so actually this is slightly different from our classrooms. We are not means we will be slightly not more formal will be there. So first there are in our body we are having number of biochemical substances, number of biochemical biomolecules we are having. If you see those biomolecules, we have different types of biomolecules, and all those biomolecules means any chemicals which is normally present in the living systems and support them in their survival for their living process. Whatever the living process on the basis of which we are going to differentiate living and non-living, that is called as the systems, the process. So like we are having carbohydrate, proteins, vitamins, minerals, nucleic acids, lipids, all these are biomolecules. Among all the biomolecules, proteins are one of the most abundant organic biomolecules in the living system. If we talk about on the earth, then carbohydrate is the most abundant biomolecules on the planet, but in the living system we are having proteins are the most abundant biomolecules. roughly 50% of the dry weight of our cells are composed of proteins only. And you cannot imagine any single function, single work in your life, in your cells, which can be performed without proteins. Think about any work, any kind of functions done by you or done by your cells. Without proteins, it's not possible. Like, think about writing, think about eating, think about walking, playing, anything, thinking. You cannot think without protein. So all the property, all the functions, all the character we are having are normally decided by proteins. So proteins are normally a functional basis, your fundamental basis you can call it, a structural as well as functional basis of our life. This protein, which normally comes from a Greek word, that it means it's proteus, which is normally it means the first place. So if you see proteins, proteins normally, if you see the functional and structural, we cannot, if you don't have carbohydrate, you can survive for some time because lipid can be working for energy source or some other purposes. If you don't have lipid, you can survive for some time, but if you don't have proteins, cells cannot survive. So proteins are having, in the terms of physiology, in terms of functions, proteins are having normally the first place. So that's why proteins, it comes from the proteus, which means the first place. And the term which was given protein, the protein term given by a Swedish chemist, his name was Berzelius. So these are some of the history. We are not too much, because that is not required too much. So now if you see the function of proteins in our body, number of functions are there, different types of classification you can have. So proteins can be classified number of bases. So most important type of classification which we are going to see, that is on the basis of its involvement in the different processes. So either proteins are the statistical, which does not normally move for their functions, which is located at one place. Normally it is a structural, like proteins present in your nail, proteins present in your hair. So those are called as statistical proteins, a static protein which does not move from one location to the other locations. So some of the proteins we are having, like collagen, alpha keratin, elastin, bone matrix. If we were having this alpha keratin is having one of the special properties. Normally almost all the proteins are functional in their tertiary or quaternary structure, but alpha keratin is a proteins which is functional in their secondary structure itself. It is a alpha helix, but it is a functional protein. So that is a speciality this alpha keratin is having. And then there are a number of other functions which is called as dynamic functions, where proteins moves from one location to the other locations, one place to the other place, one cell to other cells, one tissue to other tissue, to perform their functions. Those functions like hormones, enzymes we are having, clotting factors we are having, complement system proteins we are having, genetic proteins like transcription factors we are having, signaling molecules we are having. So they normally move from one location to the other locations to perform their functions. So this is on the basis of their functions. If you are going to see proteins elemental analysis, which type of element we are having. So normally proteins are an organic compound, and almost all the organic compounds are composed of carbon, hydrogen, oxygen. This is the three important component which is normally present in almost all the biomolecules, all the organic compound you can call it. But in our body we are having proteins which is composed of amino acids, which also contains nitrogen, because it is amino acid, so it will have amino group. So nitrogen from there, and few amino acids we are having which contains sulfur as well as. So in the proteins, these 5 elements are the main components of proteins we are having. So that is carbon, nitrogen, hydrogen, oxygen, sulfur. Other than these 5 elements, proteins can have number of other elements as well as, like it can have phosphorus, it can have iron in the conjugated forms. We will give you some of the examples also, like hemoglobin. You see, Hemoglobin is a protein which normally contains heme, and there we are having iron. We are having cytochrome oxidase, cytochrome P450. All these are proteins which is normally contains iron. We are having one of the amino acids enzymes that is called tyrosinase, that is normally associated with copper. We are having some elements which normally contains zinc, some enzymes which contain some proteins which contains magnesium, some protein contains manganese. So protein or amino acid contains different type of elements, but these 5 are the main components. Among these 5 elements, the maximum contribution is by carbon, roughly 50 to 55% contribution will be from carbon, and then we are having second place for oxygen. Now come to amino acids. What is amino acids? As its name suggests, name is having two different words. It is having amino and it is having acid. So obviously it will have amino also, it will have acid also. So if we will see the structure of amino acids, so any compound which is having a amino group and a acid group, we are going to call them amino acids. So in this compound, this is organic compound, which picture is visible to you. So this is the acid group, this acid group, and this is the amino group. So any compound which is having one acid group and one amino group anywhere in the same structure, we will call them amino acids. Location does not matter, its place does not matter, wherever it will be, but they are amino acids. So in this case we can say this is a carbon, you can call it central carbon, which is having one acid group, that is carboxylic group, and one amino group. So this is an amino acid for me. Now if you can see the place of amino groups and carboxylic groups, it can be of different locations. Like if not in the organic chemistry, if you will see, Nasir, you had a question that please show the list of topics for today's lecture. Today we will talk about proteins, amino acids, classification and its properties. Now, so on the basis of the location of amino group and acid group, amino acid can be classified into 3 categories. We can have alpha protein, alpha amino acids, we can have beta amino acid, we can have gamma amino acids. In the organic compound, whenever we are having an organic compound, numbering or the counting starts from carboxylic acid. So this carboxylic acid, they having the most priority, the first place they gets it. So the carbon which is carboxylic acid, that is going to get carbon number 1, then we are having carbon number 2, and then so on we are going to get. This is one kind of nomenclature we are having. Another type of nomenclature we are having, where we are going to do numbering on the basis of alpha, beta, gamma, delta, like this one. In case of this one, when we are talking about alpha, beta, gamma, delta, we are not going to start with carboxylic acid. The first carbon near to COOH group, the first carbon near to COOH group, we are going to call them alpha. So in this case, this is my COH group. So this carbon we will call it alpha carbon. Similarly, in this case, this is my COH group. So this is my alpha carbon. In this case, this is my COH group. So this will be my alpha carbon. So the carbon which holds, which is directly associated with COH group, that is called as alpha carbon. And near to alpha you will have beta, near to beta you will have gamma. So on you will having alpha, beta, gamma, delta, so on. Now if amino group is associated with the alpha carbon, we are going to call them alpha amino acid. Like in this case you can see, this is my alpha carbon and this amino group is associated with alpha carbon, so we will call them alpha amino acid. In this case, this is my alpha carbon and this will be my beta carbon, second one. So if the amino group is associated with beta carbon, we will call them beta amino acid. And After beta we are having gamma. So alpha, beta, gamma. So if amino group is associated with gamma carbon, we will call them gamma carbon. So we have different types of amino acids: alpha amino acid, beta amino acid, gamma amino acids. Now in our body or in the living system, we are having more than 300 plus amino acids, different types of amino acids. So some amino acids will be alpha, some will be beta, some will be gamma. But all amino acids, 300 plus amino acids, we are not going to use for protein synthesis. In the protein synthesis, we are going to use only 20 specific standard amino acids for protein synthesis. Why such? Why we are can anyone can give me answer? Why we can use only 20 amino acids for protein synthesis? Why not 300 plus amino acids? You can type in the chat box if you have any answer. Why only 20 amino acids we are using for protein synthesis? Why not 300 plus? Does anyone is having any answer? Ehtisham, Akshay, Manpreet, Nasir, Sivangi, Kanhai. Anyone is having any answer? No. Okay. Normally protein synthesis depends on messenger RNA. So which type of protein you are going to synthesize, that depends on your messenger RNA. In your messenger RNA, if this is my messenger RNA is having a genetic code. This genetic code is a triplet codon. So this triplet codon is normally recognized by your tRNA. So in our cells we are having a transfer RNA. So this transfer RNA is having anticodon which recognizes this triplet codon. So we are having only 20 different types of tRNA which can recognize those genetic codes. So we can synthesize, we can use n number of amino acids, but we don't have sufficient amount of tRNA which can recognize for all or more than 20 different type of amino acids. So we are having only tRNA which will available for 20 amino acids, and all those 20 amino acids are alpha amino acids. For our protein synthesis, we are not going to use beta, we are not going to use gamma amino acids, we are going to use only alpha amino acid for our protein synthesis. And these are some of the examples, like all our amino acids like alanine, lysine, glycine, tyrosine, tryptophan, phenylalanine, histidine. So all 20 amino acids which we are normally knowing are alpha amino acids. Beta amino acids is one of the example is beta alanine, and gamma amino butyric acid. This is one of the neurotransmitter. GABA is a neurotransmitter, that is gamma amino acids. So roughly we are having 20 amino acids. We are having few other amino acids also which is recently discovered. That is, two recently discovered amino acids are pyrrolysine and selenocysteine. Selenocysteine is an amino acid in which sulfur is replaced by selenium. Selenium is another element of periodic table. So from this in this cysteine, if sulfur is replaced by selenium, we are going to call selenocysteine. This is my 21st amino acid. And pyrrolysine, that is a lysine derivatives, which is my 22nd amino acid. So nowadays we are having 22 amino acids other than 20 normal standard amino acids, selenocysteine and pyrrolysine. Now these amino acids, the function of these amino acids are it is going to join together and give you a protein. So normally proteins are polymeric forms of amino acids. So multiple amino acids join with each other and give you proteins. So this is one of the example where this is one of the amino acids, and this is alpha amino acids. Why it is alpha amino acid? Because this is COH group, this is central carbon, alpha carbon, and this is amino group joined with alpha carbon. So this is my alpha amino acid. This is second amino acid, this is also alpha amino acids. So these two alpha amino acids joined with each other by forming a bond, a specific bond, that is, you can call it, it is a dehydration reactions. Why we can call it dehydration reaction? Because water is going out. So in this case, one amino acids carboxylic group reacts with second amino acids amino group. So this is every time whenever peptide bond will forms, the carboxylic group of one amino acid will combine with the amino group of another amino acids by removal of water. That you can call it condensation reaction also, you can call it dehydration reaction also. So they will give you a protein or a peptide. Now by convention, you can write amino acid in any way. Like I can write amino acid where COH group will be this side. But normally by convention we are not going to write like this one. We are going to write always amino group will be on my left hand side. This is convention. If you will write opposite way, no one is going to tell you wrong. But by convention we are going to write it is in the way where amino group will be on the left hand side and carboxylic group will be on the right hand side. So whenever you are going to write a peptide, protein also, that time you should remember it, amino group will be on your left hand side. And there are some basic features of this peptide bond. What is the features of peptide bonds? This peptide bond is a single bond, but because of the resonance structure, because of the chemical nature of this bond, this peptide bond is having some partial double bond characteristic feature also. So if there are number of questions coming in the competition that this peptide bond which is formed between two amino acids have partial double bond characteristic feature also. It is a single bond, but it have double bond properties. First. Second thing is, whatever we have written here, this is a cis type of peptide bond. Cis means, if you will see this O double bond tonic group O and this NH are on the same side. So this is cis structure. But normally this is not the correct way. We should write in the trans way. Oxygen and hydrogen will be in opposite side. Like here I am going to write it. So you can write like this one, C, and here we will have N and here we will have H. So this is trans way, means O will be on one side and hydrogen will be on opposite side. So this is correct way of writing a peptide bond. So mostly peptide bonds are in the trans form. Third property is, this peptide bond is a planar structure and a rigid structure, tough structure, means hard to break it, hard to dissolve it, break it. Now we are going to see the amino acids. So mostly amino acids is organic compounds, and whenever organic, we are going to see organic compounds. A special properties organic compounds are having, that is called as stereochemistry, and carbon is having tetravalency. So whenever we are going to talk about carbon tetravalency, so carbon can form 4 bonds. So like here I am going to write it. This is my carbon. This is going to form 4 bonds. So think about, just for example, here we are having A, here we are having B, here we are having D, and here we are having E. So if a carbon can have 4 different bonds and all the groups associated with them are different, like in this case carbon is having 4 different bonds associated with A, B, C, D, we can call them asymmetric carbon. So there are few rules for asymmetric carbon, asymmetricity. First rule is, carbon will have 4 single bonds, no double bond, no triple bond. So whenever a carbon will have double bond, triple bond, never it will be asymmetric. Like this is, which type of a structure is this one? What is this structure? Can anyone answer? What is this structure? Just now we have written it. This is not asymmetric, this is aldehyde. In this aldehyde we are having a carbon. This carbon is having one single bond and one double bond. So whenever a carbon will have any double bonds, means this carbon is symmetric carbon. So first rule for asymmetricity is carbon will have only single bonds. So 4 single bonds would have to be there, and all the groups associated with those single bonds should be different. Like here we can see it. Carbon is having single bonds and all 4 groups are different groups. A is different from B, B is different from D, D is different from E. So in this case, this carbon is asymmetric carbon, or you can call it chiral carbon. Whenever we are having chiral carbon, then only it can show you any kind of stereoisomers, stereoisomeric properties, or you can call it optical isomers. Now, so in this case, when we are talking about amino acids also having a central carbon. It is having one side, it is having COH group, another side it is having amino groups, then it is having hydrogen, and fourth group it is having R group. R group means it can be anything. It can be methane, it can be nitrogen, it can be methane, ethyl, propyl, butyl, alcohol, anything. Whatever you want, you can add it there. So R group can be anything. Now, in this case, this carbon, this central carbon is having 4 single bond. 4 single bond is 1, 2, 3 and 4, and all the groups associated with them are different. So this carbon, central carbon of amino acid, in this case is asymmetric carbon. So it will show me stereoisomers, and whenever we are going to call stereoisomers, we are having a formula. How many stereoisomers it will have? It will be 2 to the power n, where n is number of asymmetric carbon. So in this case, in this L amino acid structure, we are having only one asymmetric carbon, means we will have 2 to the power 1 number of stereoisomers. It means total how many? Only 2. So one is L as amino acid, second one is D amino acid. Now what is L, what is D? How to do nomenclature? How I can call it L, how I can tell it D? In the case of carbohydrate, it is slightly easy, but in the case of amino acid, it is slightly tough. How you can say it? Here we are going to do a pure organic compounds behavior, yeah, organic compound rule. So we are going to write the atomic number of the element which is associated with that one. Like hydrogen's atomic number is 1, carbon 6, nitrogen 7. So nitrogen is having 7. Now you have to go from lower to higher side. You can go from 1 to 6 and 6 to 7. So 1 to 6 and 6 to 7. So this direction of movement is anti-clockwise. So whenever we are going to move anti-clockwise way, anti-clockwise directions, we are going to call it left-handed direction, left side. It is called as L amino acid. And in this case, this is 1, this is 6, this is 7. So the direction will be right-handed side or clockwise direction. So it is called as D amino acid. So first you have to decide it is asymmetric carbon is there or not. And once you have decided it is asymmetric carbon, then we can go for it is D form or L form on the basis of right side movement or left side movement. Now this is the structure of 20 amino acids which we are having. These slides I have taken from the Baijo. So if you will see, every amino acids are having a basic structure. Basic structure means it is having a carbon, hydrogen, it is having central carbon, one hydrogen, one COOH group, one amino group and one R group. So every amino acids have this group is common in all the amino acids. The upper portion C, NH2, COH and hydrogen is common in all the 20 amino acids whatever we are having. Only they are going to differ in their R groups. So some amino acid will have R group equal to H, someone will have CH3, someone will have C2H5, someone will have benzene ring, someone will have phenol ring, someone will have different thing. So we will see those structures also. So they are going to differ only in their R groups. So on the basis of R group, on the basis of a structure of amino acids, we have different types of classification of amino acids. So amino acids are going to classified on 4 criteria. There are 4 different ways on which we can classify amino acids. The first way is on the basis of their R groups structure. Which type of R group it is having? It is aliphatic, it is aromatic, it is benzene ring, it is having phenol ring, it is having sulfur containing. So on the basis of their R group structure, we can have one type of classifications that we will see. Second type of classification scheme is on the basis of the polarity. Polarity means it is polar, it is non-polar, it is acidic, it is alkaline. So on the basis of the polarity of R group, we can classify, that is second classification. Third kind of classification we can do on the basis of the nutritional requirement, like it is essential for your body or it is non-essential for your body. If it is essential, we will call it essential. If it is non-essential, we will call it non-essential. That is called like nutritional essentiality. Fourth type of classification we are having, that is on the basis of its fate. Fate means what is the future of amino acids in our body. If amino acids are going to give you glucose carbohydrate or going to give you lipid, so that is their fate. So that is my fourth type of classification. Any questions anyone is having? If you are having any question, you just ask. है कोई क्वेश्चन किसी के पास? किसी के पास कोई क्वेश्चन नहीं है? नो वन इज हैविंग एनी क्वेश्चन? इज इट क्लियर? चीजें क्लियर हो रही है? एहतशाम समझ में आ रही है? और कौन कौन है? चलिए, आप लोगों में किसी के पास कोई क्वेश्चन नहीं है? या तो फिर सारी चीजें समझ में आ रही है या फिर कुछ भी समझ में नहीं आ रही है? चलिए, आज का क्लास बेसिक्स था. सो वी आर जस्ट टॉकिंग अबाउट बेसिक्स. तो जैसे जैसे थोड़ी हायर साइड में जाएंगे तब आपको थोड़ी ज़्यादा प्रॉब्लम होगी, थोड़ी ज़्यादा अच्छे से समझ नहीं पड़ेंगे. चलिए, सो वी आर गोइंग टू से फर्स्ट टाइप ऑफ क्लासिफिकेशन, दैट इज ऑन द बेसिस ऑफ द स्ट्रक्चर ऑफ आर ग्रुप. सो ऑन द बेसिस ऑफ आर ग्रुप, which type of R group your amino acid contains? We are having 7 classes, 7 type of class. The first class is where amino acid contains aliphatic side chain. अब what is aliphatic? जब आप ऑर्गेनिक केमिस्ट्री पढ़ते हैं तो इन ऑर्गेनिक केमिस्ट्री वी आर हैविंग एलिफैटिक, वी आर हैविंग एरोमेटिक. आई एम गुड एटिका. सो in organic chemistry we are having aliphatic, we are having aromatic. Normally aromatic compounds is having some kind of properties which we are going to call aromaticity. So on the basis of that one, we are having one class where R group will be only aliphatic chain. Aliphatic chain means it will be normal straight carbon chain, either methyl group, ethyl group, propyl group, butyl group. So normal aliphatic chain, carbon chain we will have, that is called as aliphatic chain. So the first class, there we contains 5 amino acids. Those 5 amino acids are glycine, alanine, valine, leucine and isoleucine. Now if you will see here, we have written 3 different things. complete name of amino acid, that is glycine, alanine, valine, leucine and isoleucine. Other than that one, we have also written two other things. One is 3-letter code and second one is 1-letter code. Normally, most commonly, whenever we are going to decide 3-letter code for amino acid, so the first 3 letter of amino acid is going to tell you its 3-letter code, like glycine GLY, alanine ALA, valine VAL, leucine LEU. In case of isoleucines, we are taking 2 alphabet from leucine and one is I for iso. So ILE, that is isoleucine. This is called 3-alphabet code, 3-alphabet nomenclature you can call it. Second nomenclature is 1-alphabet nomenclature. So like here we are having G, here we are having A, V, first alphabet. But there are certain abnormalities, certain kind of wrong thing happens when we are talking about 1-alphabet code. We are going to see further. Now come to a structure. So if you see the structure of glycine, I have told you all the amino acids we are having this type of a structure. So this is common in all the amino acids. So here also, in this case also, in this case also. So all the amino acids we have that structure is common. Only their R groups are going to differ. So if you will say, in case of glycine we are having H as R group. In case of alanine we are having methyl group as a R group. In case of valine we are having propyl as a R group. Here we are having butyl. Here we are having butyl only, but iso and neo forms. So these are different types of aliphatic chain. Here we don't have any aromatic ring. Only aliphatic chains are there. Only simple structure are there. So this is these 5 amino acids we are having which contains aliphatic R group. Now, so this is same thing we are having same 3-letter codes and 1-letter codes we are having. I will tell you where it is. These are the having some kind of differences. Like second kind of classification, second kind of class we are having where amino acid contains hydroxyl side chain. Hydroxyl side chain means OH group. OH group means alcohol group. So in whole 20 amino acids we are having 3 amino acids which contains OH group. One is serine, second one is threonine, and third one is, can anyone name what is the third amino acid which contains OH group? Which one is third amino acid which contains OH group other than serine and threonine? Aliphatic chain is simple structure. In organic compound we are having 2 types of structure. We are having one like carbon, carbon, like this one you can write it, or carbon, 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 like this one you can write it. This is simple carbon chain. This type of structure we are going to call aliphatic. And second type of structure we are having where we are going to write in the form of benzene ring or 5 carbon ring. So this is having a specific property that is called as aromaticity. So this is any compound which is having this type of aromatic ring are called as aromatic compound. Where we are having aliphatic chains, there we are going to call aliphatic chain. Fine. So we will talk about aromatic compound also. Yeah, third amino acid is tyrosine. So there are 3 amino acid which contains OH group, that is serine, threonine and tyrosine. Now why I have not written tyrosine here is because tyrosine is a compound amino acid which contains aromatic ring. So I have kept it in with the aromatic amino acids. We have another class which is aromatic amino acids. If you will see here, side chain which contains OH group, here serine, threonine and tyrosine. Then we are having one class of amino acid which contains sulfur atom. So we are having 2 amino acid which contain sulfur, cysteine and methionine. We will see what is the difference between the sulfur of cysteine and sulfur of methionine, how they are different. Then we are having side chains of R group which contains acid groups. They are aspartic acid, asparagine, glutamic acid and glutamine. How to do nomenclature also. Then basic amino acids. Three basic amino acids we are having: histidine, lysine and arginine. Side chain which contains aromatic ring. Here we are having phenylalanine, tyrosine and tryptophan. So this tyrosine I have not written here, I have written here. So tyrosine contains both OH group also and the aromatic ring also. And out of this 21 amino acid is there, which is not correct amino acid, it is amino acid, that is proline. We will tell you what is the difference in the amino and amino acids. So now we are going to say the amino acid which contains hydroxyl group, hydroxyl side chain. So it is serine and threonine. Again it is having two nomenclatures, three alphabet nomenclature. The first three alphabet SER and THR. In case of tyrosine, TYR is the first three alphabet. So this is three alphabet nomenclature. Now one alphabet nomenclature for serine it's S, it's okay. For threonine it's T, okay. But if I will give tyrosine also T as one alphabet nomenclature, one letter code. So how to differentiate between threonine and tyrosine? So depending on when it has been discovered. So those amino acid which was discovered first, they have got their first alphabet. So among this one, this threonine has discovered first. So threonine has got T. So obviously tyrosine we have to give some other nomenclature, some other one word code. So tyrosine we have given Y as a code. For tyrosine one letter code is Y, not T. Why? Because it should be different from threonine. So here we are having OH group and here we are having OH group. Structurally both are same only except this methyl group. So here we are having serine, threonine and tyrosine. Akshay, if you can see here we are having aromatic chain, aromatic ring, benzene ring. Is it clear? Is it okay? Now we are having another class of amino acids which we are going to call sulfur containing amino acids. So we are having 20 alpha amino acids. We are having two amino acids which contain sulfur. It is cysteine and methionine. Now you have to remember here a specific thing. We are having with the name of cysteine we are having two things, two amino acids. One is with the name of cysteine, other one with the name of cysteine. Only E is missing there. So we will tell you about this one also. Now this cysteine is the amino acid which contains sulfur and methionine is the amino acid which contains sulfur. In case of cysteine, normally we are having alcohol group, we are going to write in the form of OH. So from this OH group, oxygen and sulfur belonging to same group of your periodic table, 6th class, 6th group. So oxygen is replaced by S. So in place of OH we are having SH. It is almost same type of compound. Hydroxyl group it becomes sulfhydryl group. Sulfur is replacing oxygen, so OH becomes sulfhydryl SH group. So cysteine contains sulfhydryl group where SH is on the terminal place at the last. Whereas in case of methionine, we don't have sulfur as in the form of SH group or the sulfhydryl group. This type of group is like oxygen we are going to form ester. Similarly here sulfur is going to form ester type of a structure. So this structure, this oxygen is called as ester, sulfur ester is going to be called thioester. So sulfur is going to present in your amino acid in two forms. One is sulfhydryl and second one is thioester. So one is sulfhydryl and this is thioester. So the difference between these two structures are, these two sulfur are, this sulfur SH group, sulfhydryl group sulfur, is free to react with any compound because it is at the terminal place. So it can bind with anyone, any group, any compound. Whereas this sulfur is in between, so it is little bit protected. It is not that much free to react with any compound. So this sulfur is more reactive than this one. In our body, methionine does not have any function. Methionine is having only two functions. Either it is going to be converted into another amino acid, that is cysteine and homocysteine. That is one function, and second function is it is going to start your protein synthesis. The first amino acid of your protein synthesis is methionine. So only two functions methionine is going to perform, no other functions. Most important function I am talking about. Other function you can call it methionine. It is going to give you some methyl donors, but normally two important function is one, protein initiation of protein synthesis, second, conversion to homocysteine and cysteine. Cysteine, this first amino acid which is having a sulfhydryl group, this sulfhydryl group is normally responsible for formation of disulfide bond. How it is disulfide bond is going to form? So this is one amino acid which is having SH. This is your another amino acid which is having SH. So this SH going to combine with this SH with the removal of hydrogen and ultimately forms a bond like this one. So this is your one amino acid, this is your another amino acids. So two amino acid joins with sulfur to sulfur. This is called as disulfide bond. Now I have told you about another cysteine. This one. This cysteine is the dimer of this cysteine. Two molecules of this cysteine join together through disulfide bond and give you one molecule of cysteine. So if you will join two cysteine with disulfide bond, you will get cysteine. So that this is the reason why only cysteine forms disulfide bond. Never methionine forms disulfide bond. Why methionine does not form disulfide bond? Because this sulfur of methionine is protected. It is in between the chain. So it is not free to react with any compound, whereas this sulfur is free to react with anything. So it can form a bond. It does not form a bond. So reason cysteine forms disulfide bond, methionine does not form disulfide bond. Clear? Is it clear? Ravina, Aanchal, Komalpreet, Ramanpreet. Okay. Now, if you see the structure, so this is my cysteine, single amino acid which is having a sulfhydryl group. I have told you sulfhydryl group. And this is methionine which is having sulfur is in between, thioester group. Two molecules of this cysteine is going to join together through a disulfide bond and give you this cysteine. So cysteine is not exactly, it is amino acid, but not a monomeric amino acid. It is a dimeric form of amino acid, normal amino acid cysteine. Next category of class we are having on the basis of R group, that is the amino acid which contains acidic or their derivatives. So we are having four amino acids here. We are having aspartic acid, we are having glutamic acid. So this is aspartic acid or aspartate we are going to call. Whenever it will be ionized forms, we are going to call aspartate. Whenever it will have non-ionized forms, we are going to call aspartic acid. Similarly, glutamic acid we are going to call glutamate or glutamic acid, depending on it is ionized or it is non-ionized. Now, I have told you every amino acid is having this group is common. So here also this group is common in all the amino acids. If you see, there are groups. In the case of aspartic acid, we are having one COOH group. This is normally COOH acid group. So if acid is there, so because this acid is a extra acid, so in this case this amino acid is having two acidic structure. One is normal structure which is present in all the amino acids. Second one is a acid of their R groups. So because of two acid group, it is having acidic property. So this is a acidic amino acid. Similarly, in case of glutamate also, we are having similar, everything same, one CH2 is extra. It is also having one COH group. So both aspartic acid and glutamic acid are the acidic amino acid because they are having one extra acid group. One acid group is here and extra acid group is here. Now, this aspartate is going to be converted to asparagine by one of the oxygen is replaced with amino group. If this oxygen is replaced with amino group, we will call it amino derivative of aspartate. It is amino derivative of aspartate, so we are going to call it asparagine. Asparagine means amine of aspartate. Amine of aspartate. Similarly, if glutamate oxygen is going to be replaced with amino group, we are going to call it glutamine, amine derivative of glutamic acid. So glutamate gives you glutamine, aspartate gives you asparagine. Now come to the nomenclature. How to do nomenclature? 3 amino acid nomenclature and 1 letter nomenclature. Aspartate, first 3 letters which are got ASP. GLU, they have got it because these two are the derivatives, so it will get the another names. Now I cannot give ASP to this one also. I have already given ASP to aspartate, so I cannot give ASP to asparagine also. So we have to give something else. So we have given AS and N for amine because nitrogen is extra. So we have given AS and N for amine derivative. So it becomes ASN, asparagine, and N is for their normal nitrogen only. Similarly, in case of glutamine, we cannot give GLU. Here it will be wrongly written. It will be GLN. GL for glutamine, N for amino derivative. So asparagine ASN, glutamine GLN, and the one word letter H we cannot give G because G we have already given to glycine. So here also we have not given G. So G given to glycine, so we have to give something else. So E and Q we have given. So one word letter you need to remember if you want. Otherwise no need to remember. Name you will remember, that will be more than sufficient. So these are the 4 amino acids which is either having acidic groups or they are acid derivatives. Now there are 3 amino acids which is having a basic side chain. So we have already seen whenever amino acid will have one COOH group, that is acid group, and amine group is a basic group. So if you will see in this case, in previous case we had one extra acid groups, that's why it was acidic amino acid. In this case, this amino acid is having one extra amine group. So 2 amine group, one is here and second one is here. One acid and 2 amine. So obviously it is having extra amine, so properties will be aminic, acid basic property. So these are basic amino acid. Lysine, arginine and histidine, all 3 are having one extra amino groups, one here and in this case this is here or this one. So extra amino group is there, amine group is there, and because of that one they are basic in the nature. Now the structure which we are having in the case of arginine and histidine, they are having a special name. What is the special name is? In case of arginine, when a carbon is associated from 3 sides with nitrogen, 1 side with the double bond and 2 sides with the single bond, a carbon which is associated with 3 nitrogen on 3 sides, one side with double bond and two other side with the single bonds. This type of a structure we are going to call it guanidino group. Any carbon which is associated with 3 nitrogen on 3 sides, we are going to call it guanidino group. Second, we are having, in case of histidine, this type of ring where 5-member ring where 2 member will be nitrogen, that is called as imidazole ring. A 5-membered ring, out of this 5-member, 2 member will be nitrogen, we are going to call imidazole ring. So arginine is having a special property that it contains guanidino group, and histidine is having an important property that it contains imidazole ring. So questions coming like NEET, JRF, CSIR, UGC, that organic compound या amino acid which contains guanidino group, या how to test performed for the presence of guanidino group, then it means we are talking about arginine. If question is coming that which amino acid contains imidazole rings, then it means we are talking about histidine amino acids. So we are having 3 amino acids, lysine, arginine, histidine, which contains basic amino acids. Basic means one extra amino groups, so that's why it is basic. Then we are having amino acids which contains aromatic chain. Aromatic chain means benzene ring, not histidine. Histidine is from previous slides. So we are having 3 amino acid which contains benzene ring. Benzene ring means this 6-member ring. So it is phenylalanine, tyrosine and tryptophan. These 3 contains benzene ring. So these are aromatic amino acids. So we had, this is our 6th class. First class we have talked about aliphatic chain containing organic compound. Those were glycine, alanine, valine, leucine, isoleucine. Then we have talked about a hydroxyl group containing amino acids. There we have talked about serine, threonine and tyrosine. Then we have talked about sulfur containing amino acids, cysteine and methionine. Then we have talked about acidic amino acids, that was glutamic acid, aspartic acid and glutamine and asparagine. Then we had another class, basic amino acids. There are 3 amino acids, histidine, arginine and lysine. And this is my 6th class, which is aromatic amino acid, which contains a aromatic R group. So this is R group, and there we are having aromatic benzene ring in all the 3 cases. Out of these 3 amino acids, only one aromatic amino acid is there which contains hydroxyl or the phenolic group, means benzene ring with OH group. So phenolic benzene ring, or you can call it phenol group. So only one amino acid we are having which is having phenolic group, that is tyrosine. So whenever we are going to talk about any amino acid which contains phenol or phenolic group, that is tyrosine. Is it clear? Any question, if anyone is having, you can type in the chat box. If any one of you have any questions, you can type in the chat box. Yes, Akshay, ask. Type your question. Okay, good. And the 7th class of on the basis of R group which we are talking, that is amino acid. So normally I have told you a amino acid is having this type of a structure. They are having carbon, central carbon, they will have 4 single bonds where one will be R group, one will be H group, one COH group and one amino group. This is normal amino acid. But if you will see in the proline, in this case we don't have this conventional structure of amino acid. It is having a slightly different type of a structure. What is difference in their structure is, they are having this R group and amino group is going to form a bond. So there will be a ring type of a structure is formed between the carbon, central carbon, amino group and the R group. So we are calling it amino acid. It is not the true amino acid, it is called as amino acid. It is called as amino acid, not the true amino acid. Or you can call it secondary amino acid also. So these are 7 different classes on the basis of R group. Second type of classification which we are talking about in the case of amino acid is that is on the basis of the polarity. So we have only 4 classes and these 4 classes is visible there. I will tell you how you can remember these names, all the 20 names of 4 classification. So we have 4 groups only here. 4 groups are non-polar, polar, charged and positively charged and negatively charged. Now, because in the case of non-polar structure, non-polar group, we are having roughly 10 amino acids. So don't remember those 10 amino acids. Remember other groups where we are having small numbers. Now you are knowing that all those amino acids which is having acidic group, normally acidic group, if COH group is normally it will be in this form. If acid group is going to be charged, it will be COO minus. So normally acid groups is having negatively charged. So all those acidic amino acids, it was aspartic acid and glutamic acid, they are negatively charged amino acid because acids having negative charge. So here we are having only 2 amino acids, that is aspartic acid and glutamic acid. So you can easily, if you will remember, see the negatively charged amino acid, remember acids having negative charge and it will be aspartic acid and glutamic acid. Second class we are having that is positively charged. We know that basic group NH2, if it is going to take one extra H, so it becomes NH3 plus. So basic group is having a positive charge. So we had one classification in the previous classification was basic amino acid. There we had 3 amino acid: histidine, arginine and lysine. Histidine, arginine and lysine. These 3 amino acid is having one extra amino group. So because of that one it is positively charged here. So remember those basic amino acid. So here acidic amino acid, here basic amino acids. Clear? Now come to polar. In the case of polar groups, all those 3 amino acids, all those amino acids which had OH group, so it was serine, threonine, tyrosine, all 3 hydroxyl containing group, OH containing group, and the 2 derivative of aspartic acid and glutamic acid, that is asparagine and glutamine. So 3 OH containing amino acids and 2 derivative of acidic amino acids are polar. So serine, threonine, tyrosine because it contains OH group, and asparagine and glutamine, that is derivative of aspartic acid and glutamic acids. So that's why those 2. So total 5 amino acid we are having polar. So how many you have remembered? You have remembered 2 amino acid negatively charged, 3 amino acid positively charged. So total 5 and 5 here. So these 10 name you remember, and remaining other amino acids belonging to non-polar. So you need not to remember this one. If you remember these 10, automatically that 10 will be coming in the non-polar category. Is it okay? So you can do practice at your home little bit and automatically you can remember it. 2 acidic amino acid in the negatively charged, 3 basic amino acid in the positively charged, so 3 OH containing group and 2 amine derivative of acidic groups are in the polar and remaining is in non-polar. Fine. This was my second type of classification. Third type of classification is on the basis of essentiality. Amino acids are essential or non-essential. when we are going to call essential or non-essential depending on your body's requirement. If your body can synthesize anything depending on your requirement, like in my home I can cook anything whenever I need, so that is not essential for me. Essential is which I cannot made myself. So those compounds, those things which I cannot prepare by myself, that is essential for me. Anything which I can prepare myself, that is non-essential. So similarly in the amino acid also, in the fatty acids also, any amino acid which we cannot synthesize by myself in my body, for which we are normally depending on external sources, on the diet, on the supplement, on the food, that are called as essential amino acids. So essential amino acids are those amino acids which is we cannot synthesize by myself, for which we have to depend on others. Others means diet, food, other components. They are essential. And remaining is which you can synthesize depending on your requirement is non-essential. So we have two categories here only. Depending on essentiality, it is non-essential and essential. So how to remember that one? So remember essential, remaining will be non-essential. So for that one we are having two mnemonics. Either you can remember Matt, Will, Fly, or Private Tim Hall. So whatever you want you can remember it, and this will give you all the names like M methionine, A arginine, T threonine, T tryptophan, V valine, I isoleucine, L leucine, P phenylalanine, H histidine, Ly lysine. So these are the 10 amino acids which you can remember, or either by using this one or by Private Tim Hall. So if you remember these 10 amino acids, remaining 10 will be non-essential automatically. Now out of these 10 essential amino acids, there are 2 amino acids which we are going to categorize as a semi-essential. What is semi-essential is histidine and arginine. These two are called as semi-essential. Why it is called as semi-essential? Because when a baby is in the growing phase, in their growing stage, life means development stage, that time they normally not able to synthesize it. So in that time they are normally essential for a baby, for a growing phase, for the growing means developmental phase. But once a baby becomes adult one, that time they are able to synthesize it. So these two, histidine and arginine, when during childhood, during child it is essential, but in the adult it becomes non-essential. So that's why we are going to keep them in semi-essential amino acid. So it is semi-essential. So 8 essential, exclusively essential, 2 semi-essential, and 10 non-essential. So remaining 10 are non-essential amino acid, which is not from there. So if you remember those essentials, remaining you need not to remember it. Here histidine and arginine are semi-essential amino acid. Growing children require them in the food, so it is essential for them, but they are not essential for the adult individual because they can directly synthesize it. Now the last classification scheme is on the basis of fate of amino acids. So if amino acid is going to give either only glucose or lipids or both, so we are going to call it ketogenic, glucogenic, or both ketogenic and glucogenic. If your amino acid is going to give only fatty acids, only lipids will be given, only ketone body will be given, that is called as pure ketogenic. Only lipid will be produced, and here we are having only one amino acid, that is leucine. So leucine is pure ketogenic amino acid. It will not give you carbohydrate, it will not give you any other forms of biomolecules. Second one is ketogenic and glucogenic, which gives you both the glucose, carbohydrate as well as ketone. So they are called as ketogenic as well as glucogenic. Here we are having 5 amino acids: lysine, isoleucine, phenylalanine, tyrosine and tryptophan. So these 5 amino acids are both ketogenic and glucogenic. So 5 here and 1 here, total 6, remaining 14. So those remaining 14 amino acids are glucogenic. So they are going to metabolize and give you carbohydrate. Once it will be metabolized, it will give you carbohydrate in different forms. So whenever we are going to use amino acids or any non-carbohydrate source for carbohydrate productions or glucose production, we are going to call it gluconeogenesis. So by gluconeogenesis, amino acids gives you glucose. So these are the 14 amino acids. So this is all about basics of amino acids and classifications of amino acids. Physicochemical properties is the next, that we will talk about in another day. Will it be fine? Will it be okay? कोई क्वेश्चन है तो पूछिए, इफ यू आर हैविंग एनी क्वेश्चंस. Any question anyone is having? Nandini, how many of you understand it? जो मैंने पढ़ाया उसमें से कितनों को समझ में आई? Okay. Any question you are having? कोई क्वेश्चन है तो बताइए. No one is having any question. Good. मैं पूछूंगा. ठीक है, तो आज हम स्टॉप करते हैं. अगर आपके पास कोई क्वेश्चन नहीं है तो. श्योर साकिब, विल ट्राई. आपने मुझे पहले सजेस्ट नहीं किया था, अब मैं लूंगा ऐसे ही क्लास. Not only biochemistry, we'll talk about something else also, molecular biology also for PG students. Nandini, how many PG students are here? आप में से कितने PG के हैं? Any from PG students? एहतशाम. और कौन कौन PG के? MSc. संगीता राय. गुड. ठीक है. सो वी आर गोइंग टू स्टॉप इट हियर. सो विल सी नेक्स्ट डे. आई विल इंटीमेट यू. Molecular biology also I will take. Don't worry, Nandini. Fine. Thank you, Nandini. I will take all the classes as much as I can. आपको कुछ स्पेशल चाहिए पढ़ना तो मुझे बता दीजिएगा. I will take that class also. No, don't worry. Okay, thank you.